All right. Hello, everyone. My name is Sean. I'm an MD PhD student at the Singh Lab.
Hello, everyone. I'm Vaseem Sheikh. I'm a research associate at Singh Lab.
And today we'll be talking about our project, co-targeting ephrin receptors in glioblastoma using bispecific CAR T immunotherapy. First, just a little background on glioblastoma or GBM. GBM is probably one of the greatest remaining challenges in oncology research and care. GBM is the most common primary adult brain malignancy, and it still to this day has a very dismal prognosis with a median overall survival of less than 15 months. All patients who are diagnosed with GBM go through the same standard of care, which is surgery and chemoradiotherapy, but unfortunately, almost all patients recur, and at this point, there is no standard of care. Treatment is only palliative or to enter clinical trial. Why is GBM so aggressive? Why is it like this? Well, of course, there are many different ways to answer that question. One is presented on this slide, and it uses the analogy of a flower. If GBM is this whole flower, there's a small population of cells here in the middle. And these are like the seeds of the flower. They will branch off and produce all the different petals that make up the flower of GBM. We call these seeds BTICs or brain tumor initiating cells because they're the ones, like I said, that are seeding this flower. We think that if you direct your treatments against these seeds rather than the petals, you can actually hit GBM at the core where it really hurts and maybe improve the picture of GBM diagnostics. And one of the ways that we've looked at hitting these BTICs is through ephrins. So ephrins are a very large family of surface cell receptors on many different cells. And you can see there are many different types of ephrin receptors, many complicated signaling pathways that they go through. But the bottom line is that ephrins appear to be associated with cancer. And in GBM, they are involved in migration and invasion of the tumor to make these big bulky tumors, angiogenesis to make the blood vessels, increase in stem-like features. So these BTICs are very similar to normal stem cells in the human body, and ephrins are associated with that signaling. And ultimately, this leads to poor patient outcomes. So previous work in our lab wanted to profile the ephrin receptors on patient-derived BTICs that we have here at the Singh lab. And you can see that here. They took all the different ephrins and looked at the surface expression on both recurrent and primary GBM. And you can see that we honed in on ephrin A2 and ephrin A3. This is really shown in panels B and C. Expression of these two ephrins is a lot higher in recurrent GBM compared to primary or a normal control. And expression increases after chemoradiotherapy, so standard of care. So we wanted to explore this further, and we did some genetic studies where you artificially decrease the expression of ephrins in these BTICs. And you can see in our mouse models, when you reduce the expression of both ephrin A2 and ephrin A3, you get a much smaller tumor in these two different recurrent GBM lines compared to the normal control. And when you look at survival, there's a very significant difference in survival when the cells are not expressing either ephrin. So to come up with a therapeutic against this, we generated a bispecific antibody. Now, these days, almost everyone's familiar with antibodies after COVID. You know, you want to get your vaccine so that your body makes antibodies against targets that are on COVID. Well, you can actually engineer antibodies to be against targets on the tumor. And we engineered a bispecific antibody, which means this one antibody can recognize two targets, ephrin A2 or ephrin A3. And you can see when we treat our mice with this antibody against both ephrins, again, you see a very substantial reduction in tumor volume. However, bispecific or all antibodies do have some limitations. This includes poor biodistribution, so they're not always able to get into the tissue where the tumor is, and they have a very short half-life in vivo. So that's in mice or in people. They don't last very long when you give the person the treatment. So for that reason, we wanted to explore different immunotherapies. And that's where we had the idea of CAR T cell therapy. Now, CAR T cells are becoming very popular, but the way that they work is you take T cells, which are one of our body's immune cells, and they're very good at fighting cancer, fighting infection. They're really good killers. You take these T cells from the body and you train them to recognize and fight the tumor, a tumor antigen, a tumor target. And you do this through a CAR, a chimeric antigen receptor, which Vaseem will be talking more about later, but you use that to direct the T cell's activity against the tumor. And we've done this in the past at our lab. The first time was against CD133. This is a very well characterized marker of BTICs in human brain tumors. And again, very good eradication of the tumor and a substantial survival advantage. More recently, we've tried other markers such as CA9, same survival advantage, CD70, really good improvement. And even we tried the CD133 CAR T in a colon to brain met model, and you can see a very striking improvement in overall survival. So our objective was to, before I hand this off to Vaseem, was to engineer and test a CAR T cell that, like the bispecific antibody, can recognize both ephrin A2 and ephrin A3 and efficiently kill GBM cells. And with that, I'll pass it off to Vaseem to talk more about some of our current work.
Okay, thank you, Shan. Thanks for the wonderful introduction. Just to build upon that, what we were trying to do, what we did before was we developed a bispecific antibody that targeted both. And as Shan told, that we wanted to build a CAR T now that would target either A2 and A3. So we took that antibody and we tried to make it into a CAR T cell. So how does a CAR T receptor looks like? It can be seen in the left side of the screen. The top part is the antigen binding domain. The first red and the second red, it can bind either to A2 or the second can bind to A3, or we can switch them both. The transmembrane domain, so the antigen binding domain is like a homing device which will take the T cells to the cancer antigen and will recognize it and bind it very tightly. The CD8 transmembrane domain is like a communication channel between the outside of the cell to the inside of the cell. It communicates that, okay, now the cell has attached, and it sends the signal downstream and will send the signal to 41BB domain and CD3 zeta domain. 41BB domain helps in persistence. So fighting cancer is a tiring job. They'll get tired, and it helps them to persist more. And CD3 zeta will help in secreting the cytokines. So it is like a flipping switch. As soon as it binds, it's time to kill. On the right side, you can see a plasmid map. So this is what we use to engineer CAR T cells. And you can see the similar structure wherein and the gray is the SCFE, the ephrin A2, A3, and then there is a hinge region. There is a transmembrane domain, and then 41BB and CD3 zeta. So this plasmid will transcribe and make this receptor. Next slide. So just to go over how do we make CAR Ts, Shan briefly explained it before, but just to go into more detail, we collect the blood from healthy donors, and by a process called as leukapheresis, we isolate the white blood cells that consist of T cells, B cells, and K cells. And from this white blood cells, we enrich specifically T cells by activating them using CD3 and CD28, and that selects for specifically T cells. Now, what we showed you in the previous slide, where the plasmid map, we introduced this plasmid into a virus called as lentivirus, and then we put the virus onto the T cells. The virus infects the T cells and and the plasmid gets integrated into the genome of the T cells and then expresses this CAR receptors. And finally, we have CAR Ts, which we expand, and it is ready to test. So to begin with, we wanted to characterize what is the ephrin expression. Oh, sorry. I didn't go through so in the plasmid map earlier, there was the downstream, there was an mCherry gene that can be used to track whether the cells has been transduced or not. And so we'll see, we can see the cells through flow cytometry. They are colored, and we can test whether they have been transduced or not. And so in this slide, what we are trying to show is this in the first panel is Luke 17. That is a donor, healthy donor. And as we go into the next panel, it's ephrin A2, A3, where A2 is in the front, A3 is in the back, and we are tracking whether how many T cells contain this has been transduced with CAR, and we can see that around 24% of the cells has the CAR receptor. And in the last is A3, A2, where A3 is in the front and A2 on the back. And we can see that nearly 17% of the cells were transduced with the CAR receptor. Next, we wanted to characterize the BDIC lines for their ephrin A2, A3 expression. And we see that ephrin A2 and ephrin A3 was nearly expressed 100% on both of the lines. And yeah, making it a very good target antigen for CAR T cell therapy. Next, we moved on to testing the CAR Ts in vitro. This is very preliminary study where we use that CAR that we made, and we put GBM cells in a dish and then put CAR Ts, and then we monitor the GBM cells for killing. And as we can see that as compared to the untransduced T cells, ephrin A2, A3 had significantly higher killing, but there was no significant difference between ephrin A3, A2 and untransduced CARs. So this is very preliminary data, and we are still trying to optimize it. We are still trying to optimize the transduction as well, which we can see. Like the ongoing studies is we are trying to optimize the transduction efficiency of the CAR T cells. And we are also trying to do the in vitro cytotoxicity of the CAR Ts against multiple RGBM lines. The data shown was only of one RGBM line. And moving on, we'll try to test after optimization into. I will take this CAR Ts and test them preclinically into the mice in our patient-derived xenograft models. We would really like to thank all our partners, especially BDFC, for funding us. We would like to thank our patients, especially our patients, for providing us with the precious samples. And really thank you all. And if you have any further questions, you can reach out to Sue, and we can connect over. Thank you. Thank you, everyone.